G'day team, in this video we're going to be looking at protein structure. We're going to talk about what proteins are and how they're made of polypeptides. We'll then talk about the amino acids that make up the polypeptides and then go into the structure of protein, the primary, secondary, tertiary and quaternary structures. So to define proteins, proteins are chains of amino acids. So there's about 20 different amino acids and we'll talk about those in a second. But when these amino acids come together, they form these chains called polypeptides. Now, protein is made up of usually a number of polypeptides. There are some proteins that are only one polypeptide, but most are a number of polypeptides. And that might be a number of the same polypeptide or a couple of different polypeptides that come together to make that protein. Now, in the DNA sequence, each gene, a series of bases, codes for one polypeptide. So for a protein, you might have a couple of different genes that actually code for that protein. So the amino acids, the building blocks of that chain. So they have a standard structure. There's a nitrogen and two hydrogens on this side, a carbon, two oxygens and a hydrogen on this side. Now we don't need to get into the chemistry of that too much, but just to know that they have a standard structure. Now the interesting part is this R, because this R can be any side chain. Now when these amino acids come together, the oxygen and hydrogen and then the nitrogen on this side form this, which is called a peptide bond. And this peptide bond is where they get their name polypeptide from. So, as I said, there's about 20 different amino acids, and each one of them has their own chain on the side. So that R that we saw before could be any of these things, depending on which amino acid it is. And these different chains have different properties that interact with each other differently, as well as have different sizes. And that will be important once we get into the three-dimensional structure. Okay, so we break up the different structure or the different way we describe the structure of proteins into 4 different stages. So the primary structure is just what amino acids are present and what order they're in. So that's the specific sequence of amino acids that makes up this polypeptide. So here we see we have an alanine, a glycine, a serine, an isoleucine, and a leucine. So that is our primary structure. It is the amino acids that are present and the order that they are in. The secondary structure is a function of those polypeptide bonds. So between each one of those amino acids, there's a polypeptide bond, and those polypeptide bonds interact with each other within the amino acid. And the way that they do this is they can form either what's called a beta-pleated sheet, like this, or an alpha helix that looks like this. So these are the only 2 structures that can form in that secondary structure. And these, whether it's going to form sheets or helixes, is going to be within sections of that polypeptide. So some sections will be sheets and some sections will be helixes. When we get into our tertiary structure, those side chains on the amino acids start to become important, because as I said before, they have different properties, and the way that these different side chains interact with each other forms our tertiary structure. So some of those are going to attract each other from different properties they have, whether they're polar, non-polar, positively charged, negatively charged. So some of them will attract each other, some of them will repel each other, and some of them are just really physically large, which means that the way that the amino acids have to bend around it is going to affect that tertiary structure as well. And finally, we have our quaternary structure. So this is our last level of structure, and that's to do with how the different chains of amino acids, those polypeptides, come together to form the final protein. Now you can see here, this is a representation of DNA helicase, which is a protein or an enzyme that's involved in DNA replication. And you can see here that these different colored sections are the different polypeptides that have come together to make this protein of DNA helicase. As well as that, and this is the same protein, but another representation of it, you can see that in some sections, so looking at their secondary structure, in some sections you see the blue-coated sheets, and in other sections you see the red-coated helixes. So that's the secondary structure. So when you're looking at one of these diagrammatic representations, it gives us an idea of the three-dimensional structure. And it also gives us an idea of those structures below from the tertiary down. In this video, we've talked about proteins, which are made of polypeptides, being chains of amino acids. Amino acids which have a consistent structure that form peptide bonds when they join together and have different side groups. And then we've talked about the 4 levels of structure of their three-dimensional structure. That's the primary structure being the order of amino acids, the secondary structure being whether it is alpha sheets or beta helixes, the tertiary structure being the side chains of the amino acids interacting with each other, and finally the quaternary structure where those multiple polypeptides join together to form the final protein. Thanks for watching. Peace out.